Hello and welcome to this first video on OCR A-Level Biology. This is module 2.1, which is cell structure. Now this is the first topic you need to know before doing anything else in biology. And the first thing that we need to be aware of is to think about, where's my pen? Oops, okay, one. Is to think about scale. So if I just write it here, so we need to start first of all talk about scale. And scale is pretty important in biology. So which is why I have this. So we need to zoom in on here. This is like yeah, I've got a new setup. I just got started using this new setup, so I don't want to be edited. So our human eye can see, with our naked eye, you can see up to 0.1 millimeters, which is roughly around 100 micrometers. But it's possible to see further than that. We use something called magnification. So if I do this, so there's two types of microscope. There's this red one, which starts all the way here, which has limits between this point here and this point here. And then there's also this blue one, starts at this point here and ends all the way up here. It's not really exact, but I'm just getting used to it. So the red one, first thing, the red one is called a light microscope. The blue one, as you can see there, this is and they were and the light microscope were the first types of microscopes invented like the late 1600s. But more recently, we've discovered and invented a new type of microscope called the electron microscope. And light microscope, as the name suggests, uses waves of light to produce an image. Electron microscopes use beams of electrons to produce an image. The advantages of using a light microscope is, well, you're able to see living specimens. But however, light microscopes have a limit. They have a limit of up to 100 nanometers, so you can't see anything smaller than 100 nanometers. So you can't see this thing, viruses, or you can't see like individual molecules or individual atoms even. You can't see that with the light microscope. Therefore, the electron microscope was invented to look at things even smaller than cells. So look at the individual proteins, individual viruses, individual atoms within cells. The only disadvantage of electron microscopes is that, one, they're really expensive to use, and two, their specimen has to be dead. Light microscopes, they're cheaper and they're portable, which means you can take them out if you are doing some field work. You can take these microscopes with you. They're relatively easy to use. For electron microscopes, you have to do a lot of intense training. And plus, they can be, and the most important thing is they can be viewed as living specimens. So now we're going to go through this scale, if I can find my button there. And we're going to start looking at the different types, different things, objects you can see by using each of these these microscopes. So the first thing that we have here is an individual atom, and you can only see these things using electron microscopes. So this first one is an atom. An atom is the smallest non-breakable object or thing in the universe, basically. Obviously you can break it down into like protons and neutrons, but we'll get to that in chemistry. Then we have phospholipids. So phospholipids, these make up the cell membrane. We have a whole topic on the cell membrane later in module 2. And these are around 1 nanometer. Then we get to 10 nanometers and you start to see certain organelles and also proteins, certain molecules such as proteins. So this organelle here is called a ribosome. And you can also see proteins. So we have another thing. And then we come down to here, and then we have here at 100 nanometers, where you're just not able to see this with the microscope. You can only use an electron microscope for this, and then these are viruses. So this one typically is the flu virus. And then we start getting into the light microscope range. So these objects you can see with both an electron and a light microscope. We tend to use light microscopes in schools and at universities. And here you can see certain organelles. We tend to use electron microscopes for organelles like mitochondria, Golgi apparatus, chloroplasts, because they're much easier to see. Light microscopes, you would only be able to see the nucleus, cytoplasm, cell membrane. That's pretty much it. But you can try and see, but it's a bit difficult. We tend to use electron microscopes for these. So these ones are mitochondria. I'm just going to write it mito for short. These ones are chloroplasts. I'm just going to write that for short. And then these ones are prokaryotic cells, which we'll get to later. And these are bacterial cells. We then start going into around 10 micrometers. And then here we get to see whole cells. So these ones are cheek cells, human cheek cells. And then this one is an egg cell or ovum or oocyte. Because at university we don't call them ovum anymore, we call it oocyte. And then we start getting into past 100 micrometers, and then we start getting into around a millimeter. So just below a millimeter, we see these cells which are onion epidermis, if I can spell that correctly. And then if I just move down, so it's easier for myself to write. See if I do it there. And then here, at 1 millimeter, we can just about see this with the naked eye. This is amoeba, an amoeba cell. And then finally, here at 10 millimeters to 100 millimeters, we have a chicken egg. We can clearly see this with the naked eye. If we were to view a chicken egg under the microscope, it would look ridiculously big. So there we go. So that's all to do with scale. So if I do this, recenter view, if I just do this before, and now let us go down. There we go. So now we talked a bit about scale, so I'll just show you this quickly. So as I said, there's two types of microscope, electron and light. Let's just write a few more bits about them. So electron microscope, I'll just write one sentence right now, use beams of electrons. And here also specimen has to be dead specimen. They were used to use for dead specimens. Light microscopes, I'm not going to go into too much detail about these two types of microscopes. If you want more, go to the textbook. I think it's the first page, I think, in this module, in this topic. Here we use light waves. waves of visible light waves. And then here they can be used to view living specimens. So that's just a bit more information about them. But as I said, go to your textbook if you need any more information. So I've just got here some plant cells under an electron microscope. So this was actually a question in the textbook, and it asked you to label what A, B, C, A to each were. It will sometimes ask you to calculate the actual width, the actual diameter of this cell here, WX, and of this actually this isn't the cell, this is a nucleus, of this nucleus here, and it will ask you to calculate the actual width of this organelle and this organelle. To do that, we need to use the magnification formula. So I just scoot over here for a second, and then zoom in. Then look at the magnification formula. Okay, so magnification is how big you can, how many times bigger an object is under a microscope. We have two terms to do with microscope: magnification and resolution. I'm not going to go into them in more detail, but magnification is basically how many times larger an object that appears under a microscope compared to its actual size. And resolution is basically how clearly you can distinguish between two objects. So say if two objects need to be like 0.2 micrometers apart to be clearly distinguished as two objects, then the resolution for that microscope is 0.2 micrometers. But I'm not going to go into much more detail. Here, so actual size is A, I is image size, M is magnification. So if I wanted to calculate actual size, I would cross off actual size here. Then I'll be left with I over M, which is image size over magnification. And that is a very bad M. If I want to calculate magnification, if I wanted to calculate magnification, sorry, for the first time, I will cross out M, and then I will be left with I over A, or image size, over actual size. So there we go. Actual size is image size over magnification. Magnification is image size over actual size. And that's pretty much all you need to know from there. So now we're going to look at plant and animal cells and the different structures located within plant and animal cells. First up, we'll look at animal cells. So this here is an animal cell. And animal and plant cells have quite a few in common. They have quite a lot of organelles in common. But plant cells have different organelles, with some extra organelles as well, but we'll get to that later. So animal cells. So the first thing you can see here, this purple bit, is the nucleus. The nucleus, I'm not going to write it here because I don't have much space. The nucleus is basically contains all of the cell's genetic material, basically DNA, and it's where the first step of protein synthesis, transcription, occurs. So in transcription we have mRNA polymerase making that transcript of messenger RNA from a specific gene within the DNA, and then that mRNA will leave the nucleus and head on to do the next step of protein synthesis. So within the nucleus we also have a space called the nucleolus, and this is where ribosomes are produced. basically. That's all you need to know about the nucleus. And then surrounding the nucleus we have a nuclear membrane. This nuclear membrane has pores to allow substances like mRNA to exit and enter the nucleus. Okay, so attached to the nucleus, and this whole thing, nucleus, nucleolus, nuclear membrane, this whole thing is classified as the nucleus, basically. But the nucleolus is in the middle. The nucleus has a membrane around it, the nucleolus does not have a membrane around it. Often in diagrams it is shown with a membrane around it, but in reality it doesn't. So here, just attached to the nucleus membrane, we have two types of endoplasmic reticulum. We have smoothia and roughier. So this here is smoothia. And smooth endoplasmic reticulum is basically responsible for lipid synthesis. It's smooth because it's smooth. There's nothing attached to it, unlike the roughier, which we'll get to later. But the smoothia is mainly responsible for lipid synthesis. And that's all you need to know about it. So we also have roughier, which is here, rough endoplasmic reticulum. ER stands for endoplasmic reticulum. And roughier is where it's called roughier because we have ribosomes. And ribosomes is where the second step of protein synthesis will occur. We're actually taking that mRNA and using it to build the protein using the ribosome. And that's in translation, that step is called. And then once those proteins are made, they'll be sent from the ribosome, from the rough ER, all the way down to here, the Golgi body. And the Golgi body is responsible for packaging, modifying proteins, modifying lipids. It's basically a modification and packaging center, the Golgi body is. And then once those proteins are done, they'll be sent over to the cell surface membrane or plasma membrane to be excreted if they need to be secreted by exocytosis. We also have some little other organelles here, which have nothing. We also have other organelles around here. So these pink blobs you can see, these are called lysosomes. And quite a few cells have them. If this was a white blood cell, these would be. If this was a white blood cell, if this was a white blood cell, these would contain digestive enzymes and starting to break down foreign matter, such as pathogens or any old bits of organelle, mainly pathogens or other foreign matter. And then over here we have mitochondria. And these are basically with the cytophobic cellular respiration. And that's it for animal cells. And so now we're just going to move over to plant cells. And plant cells, you can see here, as you can see here, they're quite similar. They have a lot of the same organelles. So you still have the nucleolus. You still have nucleus, and you still have nuclear membrane. You still have, I think that's the roughier, so I'll write aria for short. You have the smoothia. You have ribosomes. You still have cell membrane or plasma membrane. You still have mitochondria up here. So let me just label all of this. And you still have cytoplasm, which I didn't label there. So all of these organelles within here are held in a jelly-like substance called cytosol or cytoplasm. So where's the relabel cytoplasm here? We just work out. I'm just going to label it here. Cytoplasm. And they still have cytoplasm. A plant cells has some different organelles. And we're going to label those in green. Because green for plants. So the first big difference, the major difference, well there's two major differences, is one, these green fins here. So these green fins are called chloroplasts. And these are responsible for photosynthesis. So they are where the photosynthesis will occur. This is where photosynthesis will take place, taking carbon dioxide and water and using it to produce glucose and the byproduct oxygen. You also have here a vacuole that contains cell sap and it helps maintain osmotic pressure within the cell. We also have here a cell wall, typically made out of cellulose. Bacteria do also have cell walls, and so do fungi, but their cell walls are made of different substances. And that's also used to maintain like turbidity and make sure the cell doesn't burst when it's in a very in a very water-like solution. So when there's lots of water in the cell's environment. And then we have here this last one is called amyloclast, which is basically starch grains. So you can see the difference between a plant and an animal cell. So this was a plant cell and that was an animal cell. So let me just go back to black. Okay, so we look at some differences between plant and animal cells. And I am doing so much. Okay, so now we're going to go into a little bit more detail of some organelles. So if I just zoom in here, this is the mitochondria. So this is the mitochondria. I'm just going to quickly label this and then we'll move on to the chloroplast. The mitochondria, we have here, there's a few parts of the mitochondria. We'll come back to this in year 2. So this is here, I think that is in there, the mitochondrial matrix. I won't go over what these are for because you'll learn more about them in year 2. And then these folded the mitochondria have two membranes. They have an outer membrane, which is this purple bit here. And they have an inner membrane, which I believe is this arrow showing, inner membrane. I believe it's showing that. Hold on 1 second. Let me just double check. Inner membrane. Yep, this is inner membrane here. And then here I think this is the intermembrane space. And then within the inner membrane there's these folded bits here of the inner membrane, and these are called cristae. So that's all you need for the mitochondria for now. And this here is just an electron micrograph of the mitochondria. Oh, I've also forgot to mention there's different types of electron microscopes. So I'm not going to go into much detail because of time. But yeah, there's more in your textbook about the different types of electron microscopes. Excuse me. So now we move on to the chloroplast. So here, up here, this is the outer. They also have an outer membrane, and they have an inner membrane, which are labeled on here, inner membrane, and this is the intermembrane space. We also have here called integranal lamellae, and these things here are called thylakoids. And lots of thylakoids make up one granum. So this is one granum. and then the fluid space that's surrounding these grana and intergranal lamellae and thylakoid is called the stroma. And that's really all you need to know about the mitochondria and chloroplast for year 1. And we also have endoplasmic reticulum, which I'm not going to go into, but there's more here. But I will show you some of the key organelles here. Key parts. So this bit is smoothia, and then this bit here is known as rough ear. And then here we have the nuclear membrane. So this is the nuclear membrane. And then attached to it is the aria and SCR. And then attached to the aria, as we said before, is our little units called ribosomes. And this ribosome is a bit weird. Now the aria and SCR do have their separate little features, but I'm not going to go into that much detail because, yeah, if you want to have more details, just go to the textbook. Because this video is really a review of the whole topic. So now that we looked at the different organelles, we are going to look at how those organelles work together to perform a certain function. This function is called, this particular one is protein synthesis. So in step 1 of protein synthesis, the DNA has to be transcribed into mRNA by an enzyme called RNA polymerase. So here we have our DNA, and then this is the specific gene that needs to be transcribed to produce a protein. Oh, and then this here is the and then this will produce mRNA will transcribe this gene and then turn it into mRNA. So this is our mRNA. mRNA would then leave the nuclear membrane, will leave the nucleus via the nuclear membrane by these ports, and then travel to the rough endoplasmic reticulum, where there are ribosomes attached. And then this is where transcription will occur. So transcription will occur here. No, not transcription. Sorry, transcription occurs in the nucleus. Translation occurs in the at the ribosome. So sorry about that. Let me just get rid of all of that. So translation will occur at the ribosome. Yeah, it is very hard to get the two confused. We have a whole nother topic in this module about DNA, RNA, transcription, translation. And then that would be used to produce a protein. And this protein specifically, this textbook's using, that protein is called insulin. So this is the insulin protein. So this insulin protein will then travel in a vesicle which is pinches off the rough endoplasmic reticulum towards the Golgi apparatus. So this here is the Golgi body, where here it enters through one face of the Golgi body and exits through the other face. So this, you don't need to know the names of these faces. You have cis face, trans face. So I don't know which one it enters from. I believe it's the cis face it enters and the trans face, but you need to double check that. I need to double check that as well. And then once it's left the Golgi apparatus, it will travel in a secretory vesicle. So this is a secretory vesicle. And it will leave the cell through the cell membrane or plasma membrane via exocytosis. Nope, not aeocytosis. with exocytosis. Okay, and that's a brief overview of protein synthesis. So now we're going to move down and look at the differences between prokaryotic and eukaryotic cells. But before we do that, we need to look at prokaryotic cells. So this is a typical bacterial cell, and at first glance you can see it's quite different from a normal human eukaryotic cell. Well, for starters, there's no nucleus. So there's no nucleus in a prokaryotic cell, but they do still have DNA, but it's held in a nucleoid. A nucleoid is just a space within the cell, within the prokaryotic cell that contains all of the DNA. We do still have ribosomes. And we still have cytoplasm. We do still have ribosomes. And we do still have the cell membrane, which is over here. But we also have some extra features of prokaryotic cells that eukaryotic cells don't have. So for example, I'll write this in I'll write some code. So for example, we have plasmids. And plasmids are basically like extra DNA. And I'll do a little bit more about them. Then, if you want to read out more, they contain extra, not essential genes, which will be normally contained within the nucleoid, but they have like extra genes. And one of those extra genes is for antibiotic resistance, which is why antibiotics, when trying to treat some bacterial infections, can be challenging, is because antibiotic resistance, and because bacteria can transfer these plasmids between each other, so they can basically share, in a process known as conjugation, they can share these plasmids with each other. And if the plasmid happens to have an antibiotic resistance gene, basically the bacteria who already have this plasmid, which contains the gene for antibiotic resistance, is just passing it on and like sharing it with other bacterial cells so that they can have resistance to antibiotics. And normally that gene transfer using of plasmid is done by little filaments called pili. Pili, I don't know how you say it. I think it's pili in the UK, and then Americans call it pili. But I'm not sure. It's probably pili in the UK. We also have bacteria, also have a cell wall, as you can see here. But this cell wall is made out of peptidoglycan, unlike plant or fungal cell walls, peptidoglycan. And then surrounding the cell wall, bacteria also have a slime capsule. A slime capsule just for extra protection. And then here we also have flagella, which help with movement. Flagellum. Flagellum is singular, flagella is plural. So before we go, so that's basically the whole module. There's one thing I haven't talked about, which is the endosymbiosis theory. It's not really essential, but it would be helpful to know for your exam. So, yeah, endosymbiosis theory is basically because mitochondria and chloroplasts, because they have their own DNA, they are thought to have evolved from prokaryotic cells, which were swallowed up by certain eukaryotic cells, and then they had a symbiotic relationship with each other. So before we go and finish off, let's do a quick comparison between eukaryotic cells and prokaryotic cells. So I'm going to start with eukaryotic cells first. The first thing is that they have a nucleus, and that's probably the biggest difference between eukaryotes and prokaryotes. And they do have membrane-bound organelles. They also are much larger than the prokaryotic cells. You can still see prokaryotic, so they're more able to be seen clearly under a light microscope. You can still see prokaryotic cells under a light microscope. I mean, you can, but it's probably a little bit difficult, so you'll probably most likely need an electron microscope. But you can still see prokaryotic cells under a light microscope as well. So now we have eukaryotic cells. They have larger ribosomes. So another difference between these two is the ribosomes. So these ones have ATS ribosomes, I believe. I think they do. ATS ribosomes, these ones have. And for plant cells, their cell wall is made out of cellulose. And then if I move down a bit more, because of fungal cell walls, because fungi have cell walls too. Fungal cell walls. They are typically made of something called chitin. So now we move on to prokaryotic cells and their differences. What makes prokaryotic cells unique from eukaryotic cells? So they have no nucleus, that's the biggest difference, and no membrane-bound organelles. They're generally much smaller, and they have smaller ribosomes as well. So, oops, I did not mean to say small ribosomes, I meant to say 70, they have 70S ribosomes, these ones have. They also have a cell wall made out of peptidoglycan, or pseudopeptidoglycan. The reason I say pseudopeptidoglycan or pseudopeptidoglycan, the reason I say that is because archaea also have cell walls, and they're also prokaryotes, but their cell wall is pretty different to bacteria, and it's basically one of the differences why archaea are separated from bacteria, or pseudopeptidoglycan. You don't need to know this stuff about archaea, it's mainly bacteria. So I'll just put it pseudopeptidoglycan. I spelled this so wrong. So cell walls of bacteria have peptidoglycan, and archaea has pseudopeptidoglycan. And that's pretty much it. So that's it, so guys, that's it for this video. And I thank you all very much for watching. So hope you found this video useful and enjoyable at times. And I thank you very much for watching, and I'll see you in the next video. Goodbye.